which comes under SIPI, Signal and Image Processing Institute, which is well known quite all over the world. So Justin got his PhD from Urbana-Champaign in Italy, and his advisor was Zhipei Liang. So little side history here. Zhipei Liang, he wrote this famous textbook on MR signal processing, along with Paul Lauterbur, who won the Nobel Prize in Medicine for the invention of MRI. And Justin was present at these ceremonies at Champaign here. So Justin has won several awards throughout his career, including the NSF Career Award, and he's also the associate editor for the IEEE Transactions in Medical Imaging. So Justin does very interesting work on MR acquisition, some physics, signal processing. So today his talk is going to be about how to exploit low-dimensional information from MRI to make it faster and better. So in other words, make MR acquisition great again. So let's welcome Justin.
Well, thank you for the very kind introduction, and thanks also for the invitation to come and speak here today. So here's a quick outline of what I'll be talking about today. I'm going to give you a quick introduction to MR. I think a lot of people in the room know MR already, so if I'm talking about things you already know, I hope you'll bear with me on that. But I'm going to talk about the opportunities and current limitations of the technique, and then I'll switch and start talking about some of the curses and blessings of dimensionality and how we can use that to address some of these opportunities and limitations, and in particular to achieve faster and better MRI. And I'm going to show you a few different examples of how that works. And then finally I'll wrap up with some conclusions. So MR. So I think a lot of you in this room are familiar with MR. It has a lot of nice features. It uses a very strong magnetic field. They don't make safety videos like this anymore. This is a safety video from the 90s that, so don't do this, but this is illustrating the power of the magnetic fields that are involved here. But there's no ionizing radiation, and so it's very much a safe imaging modality. You can go very deep inside the body. You can see basically every piece of the body you want to see. You don't have to inject anything to be able to see interesting things from the body. There are these nice intrinsic contrast mechanisms. The physics here is really, really cool. And we also have tremendous flexibility, and I'm going to illustrate that here on the next slide. There's this nice quote from Richard Ernst that says, "There's hardly another technique that is so informative for so many different types of applications." If we compare MR to other modalities, it's not necessarily that MR is better than any of these other modalities at the specific thing these other modalities are really good at, but MRI is a good generalist. It does lots of things very, very well. People are probably most familiar with anatomical imaging, but we can see lots and lots of different aspects of what's going on inside biological tissues with a number of different lenses through MR. And while it's a powerful technology, it faces several obstacles. And there's this set of obstacles that I call the Bermuda Triangle of imaging. I used to call that the Devil's Triangle, but Brett Kavanaugh sort of ruined that for me. So we'll call it the Bermuda Triangle of imaging for right now. We have this trade-off, and it's a trade-off that's common to most imaging modalities, where you can trade signal-to-noise ratio for resolution and imaging speed. So if you want to make one of these good, you usually have to make compromises in one or both of the other aspects. And just as an illustration of how this works, here's some diffusion data from an ex vivo mouse brain. If I spend a long amount of time acquiring data, and if I acquire high resolution data, I can get a pretty nice looking image here in about 30 minutes of data acquisition. These times aren't necessarily completely accurate, so this is sort of a retrospective modification of the actual experiment. But the relative values are all correct. If I wanted to get the same spatial resolution, but I didn't want to spend that much time, I do less data averaging, and I get an image that looks like this. It's clearly a lot noisier than that image there. Alternatively, I could go with a low spatial resolution acquisition, and I'd get an image that looks like this. The SNR is high here, but we've lost a lot of the spatial features that we'd see in that image. And so this trade-off is undesirable, because ideally we'd want to get this quality of image in that amount of time, but we have to grapple with these sort of fundamental trade-offs that exist. And an important thing to know is that these trade-offs get worse as you go to higher and higher dimensions. So why do we care about high dimensions? Well, it turns out that the higher and higher you go in dimensions, the more you're able to see about what's actually going on biologically. So the 1D image here doesn't tell you too much. The 2D image tells you quite a bit more. 3D, of course, is probably what you're most familiar with. But then if you're used to looking at these 3D brain images, for example, it may surprise you to look at what happens if you do some time-resolved imaging. The brainstem is in there bouncing up and down in all of us right now. This is not someone with a pounding headache. This is just a normal person whose heart is beating. The brainstem is bouncing up and down dynamically, and you wouldn't know that if you just looked at the 3D image. And a lot of expert neuroscientists I show this to are sort of blown away by how much actual movement that there is in the brain, because they don't know, because they're looking at lower dimensional representations. So high dimensions are nice. They give us more information, but there's a problem associated with that, and this is really the curse of dimensionality. That as I go from one dimension to 2 dimensions, I've basically squared the number of samples I need to take if I want to fully encode that space. If I go to 3 dimensions, it's now cubed, and so the required number of samples is basically growing exponentially in the number of dimensions that we have in our experiment. So let's think a little bit more about high dimensional imaging. So we've already said that 3D imaging, so this is a 2D slice of a 3D image. 3D imaging is useful. You can see all kinds of wonderful things in this type of image, but adding dimensions gives us more information content. So if we add a time dimension, a functional dimension, we are able to see more. If we add a different type of time dimension, we're able to see more. If we add a spectral dimension, we're able to see more, where the peaks here in this spectrum correspond to different brain metabolites. We can get a nice window into the biochemistry of the brain from that. If we add 3 or more diffusion dimensions, we get extra information from that. If we maybe inject a tracer, we can start getting some extra kinetic information from that. And if we start doing modifications of our sequence, we can get relaxation information from that as well. So in principle, if I wanted to add all these dimensions up, I'd get this super informative experiment, but we're at 3, 4, 5, 6, 9, 12 dimensions. That's not really going to be practical for us to encode all of those dimensions at once. And so mostly what people do is they'll choose a small number of these to do in a single experiment. But it would be nice if we could do all of them at once, because we'd really get a lot more information from that type of exam. So this requires us to be able to go faster with our MR experiment. And so MR's been around for a long time, and people have been working to make MR faster for the same amount of time, because the slow speed has always been a limitation of the technology. There are a variety of different approaches. There are physics-based approaches. There's approaches based on buying or developing much better hardware. But the approach that I'm going to talk about in this talk is based on signal processing, where we say we know something about our image in advance, and we use that prior information in order to do a better job of reconstructing the data we've acquired, but then also this gives us a new avenue for how we acquire data in the first place. So this is sort of a new way of thinking about the MR experiment. So if we look at the traditional MR paradigm, we'd start with some kind of data acquisition. The data looks something like that, and then you go through some sort of standard image reconstruction. But the people in this room will know we don't actually stop there, right? So that image is not the end goal of the MR experiment. That image will then get used in a variety of different ways depending on exactly what you're trying to do. There can be radiological interpretation, you can put that in a database, you can do some quantitative things with it, etc. Traditionally, the way MR works, all of the information we learn through these processes doesn't actually get fed back into the way you do data acquisition. But what I'm going to describe in this talk is taking some of the information you might be able to learn from all of these end goals and using that to sort of close the feedback loop so that we can design this data acquisition in a nicer way and get better images with less time. So, and the way we're going to do that, we're going to rely on the blessings of dimensionality. And there are a variety of different ways I could describe the blessings of dimensionality. There are some very technical mathematical ways of stating some of the blessings of dimensionality. The basic idea is that if you see random structure in high dimensions, it turns out to not be quite as random as you might initially suspect. That there's this thing called a concentration of measure phenomenon, et cetera. This is too complicated for this talk, so let me give you a nice intuitive example of this. We already know in our daily lives that high dimensional data sets are pretty simple. And an example of this might be standard images. We all have digital cameras or phones that have digital cameras in them, et cetera. When we take an image with that digital camera, there are a large number of bits there in that image. A large number of pixels. Each pixel has a large number of bits associated with storing the RGB value that's present in that pixel. And so in principle, if you have megapixel, gigapixel, however many pixels you want, in principle, that's a lot of data that you would need to store. But in reality, these images are simple enough that methods like JPEG compression work really, really well. I can take this squirrel image here and compress it down by a very significant factor, and you can't tell the difference visually. And it's because this image is not arbitrary, it's not random noise, it has structure that is sort of inherent to the fact that it's a natural image of the natural world. And we see this not just with images, we see it with audio, and you can think about MP3s. We see this with videos, and you can think about the various video compression formats that get used there. And even just generic data, we know that zip compression works really, really well for taking a large data set and representing it in a small way. That's because there's sort of this low dimensional structure that's hidden in the data. We just need to sort of figure out what that low dimensional structure is so we can exploit it. And the same thing is going to be true of MR images. And so this sort of brings us to the overall framework that we use in my group for constrained imaging. And there are a lot of different ways constrained imaging can work. I'll show you a few different examples of this as we go through the later parts of my talk. But the basic idea is we start with some kind of context-specific prior information. Lots and lots of different contexts, so there's a lot of different ways this can take shape. But then given that prior information, we use it to build an imaging model. And there are a few different aspects of this imaging model. On the one hand, there's sort of a physics model that we can put into there. So we don't have to rely on simple approximations if we don't want to, right? There's a time and place for approximation. We can use those approximations if we want to, but we don't have to. But then at the same time, we're going to make additional assumptions that capture some of the things we know to be true or suspect to be true about MR images. And then once we have that imaging model, we're going to use that to do two different things simultaneously. On the one hand, we're going to use our model to change the way we do data acquisition, and at the same time, we're going to use that model to change the way we do image reconstruction. And these, of course, are linked together, right? You can't really separate those two from one another. So in order to sort of get to the bottom of how all this works, let me tell you a little bit more about MR physics, that at least in its simplest form, the data model for MR is roughly a Fourier transform. So you have an image here, and the data you acquire is basically the Fourier transform of my image plus some noise. That is, we don't actually measure this image directly, we measure something that looks more like that, and then we apply some kind of signal processing to go from this type of image, to go from this type of data to the types of images you're used to looking at. Given that this is a Fourier model, that sort of tells us some conventional ways of how we need to acquire data. And in particular, if we think about classical inversion of the Fourier transform, the theory tells us that there's this nice Whittaker-Kelnikov-Shannon sampling theorem that tells us, hey, we need to sample this at a rate governed by the Nyquist rate. And so that sort of says, you need to acquire all this data in order to reliably reconstruct this image. But when you look at the sampling rate required by Nyquist, that can be burdensome, right? That can be a lot more data than we actually want to acquire, because acquiring data takes time for us. And so rather than that, it would be great if we could just, say, acquire a few of those data points instead of acquiring the whole thing. But if we were to do that, we need to come up with a different way of reconstructing our image. And this is where model-based reconstruction comes in, that if we can come up with a nice model for my image that has a small number of degrees of freedom in it, then I only need to make a number of measurements in proportion to the number of degrees of freedom. I don't need to necessarily follow the Nyquist rate anymore if my model is simple enough that a few data points can capture all the structure that's present there. So that's sort of the basic overarching view of the way this will work. I'm going to walk you through a few different examples of this for different types of prior information for different types of imaging models. And these are the four I'm going to tell you about today. I'm going to spend more time on the first and the last than I'm going to spend on the middle ones, but you can always ask questions if you want to know more. So the first one I'm going to start with is related to shared edge structure. So shared edge structure really comes into play when we're talking about acquiring sequences of images. And acquiring sequences of images is very common in a lot of different applications. So the examples I'm showing here are largely related to quantitative MR experiments, where we acquire a series of images of exactly the same anatomy. We've varied something in the pulse sequence to encode some contrast information that we're interested in, and then maybe we want to use that set of images to estimate a relaxation parameter or estimate a diffusion parameter, etc. So the fact that these images look at exactly the same anatomy, just under slightly different perspectives, is really powerful for us, because there's a huge amount of redundancy between this image and that image, if you know how to exploit it. Now, these types of images are widely acquired, and if you've acquired data in this way, there are a few different ways you can use the fact that these images are correlated with one another. On the one hand, you could use it to do sparse sampling. So rather than sampling all of our Fourier space, maybe we're just capturing a few different points in the Fourier space to reconstruct an image. But another application is denoising, right? So sometimes there are applications where you're really SNR limited, and you have to do lots and lots of data averaging to get a good result. In those cases, you can use this type of constraint to reduce the noise without taking the extra time to actually do data averaging. And so we've worked on both of these. In this talk, I'll be focusing more on the denoising aspect because I think some of our results there are pretty nice. So let's dig into this a little bit further. If we look at MR images, there are a few things that we can say about them. Basically, they're mostly smooth, except where they're not, right? They have mostly smooth features, but then there's a lot of edge structures in them. And if we look at different images of the same anatomy, there's a tremendous amount of correlation in the edge structure that they possess. So I have sort of scatter plots down here of the correlations between the edges here, and you can see very, very linear. If I compute the correlation matrix, it's really, really correlated. And the fact that it's very correlated means that there should be something we can exploit here to say, well, I don't necessarily need as much information about this image if I already know something from that image. So the way we exploit this is a little bit mathematical. I'm going to try not to go into too much detail here. We're going to model this set of images using something called a compound Markov random field. The basic idea is we're going to assume that within any single image, the pixels that are next to each other, the voxels that are next to each other, are mostly smooth, right? That there's going to be strong similarities if you're close together, but the amount of similarity is modulated by a shared edge structure that is present. So here I have the series of mouse images, and in red I have the same edge structure imposed on all of them, right? And so that there's this shared edge structure that exists between all of them. And then I have sort of this potential function that says we can declare edges with some level of rarity, right? Edges are not everywhere, and that's what makes this sort of a valuable constraint. Now, I'm skipping a huge amount of mathematical detail here. So this is very much not a complete description of what we're doing, but the basic idea is we're going to be trying to model this shared edge structure in such a way we can exploit it in reconstruction. So if we go back to our framework, we're taking context-specific prior information that we have this sequence of images that are really, really related to each other. You can see how related they are by looking at this video. And we've used that to build an imaging model. And our imaging model basically says, well, we have each of our different images, but there's a shared edge structure that's present between them. And we're going to want to take that and use that to modify the way we do data acquisition and modify the way we do image reconstruction. On the image reconstruction front, there's mathematical algorithms and such that we're using to exploit this model. I'm not going to go into the details of that for this talk. On the data acquisition side, we have some interesting observations. And on the one hand, our imaging model tells us, hey, you don't have to average as much as you used to average. You can still get good results. But we have theory that gives us very precise understanding of sort of the signal-to-noise ratio and resolution trade-offs that we're making by imposing this kind of model. So one of the problems with model-based imaging is what happens when your model is wrong. And so we're resilient to that in this case because we have a strong theory that captures what's going on here. And actually our theory for this SNR resolution trade-off contradicts some of the classical understanding in the field on how you should trade off resolution and signal-to-noise ratio. So classical wisdom in the field says that if you're interested in structures at a specific spatial resolution, it is wasteful to acquire images with higher resolution than that. This goes back many decades. That's the common wisdom, that you don't need to acquire data with higher resolution than what you actually care about. But what our analysis shows here is that actually it's beneficial to acquire a little bit higher resolution than you need, and then use smoothing, spatial smoothing, to go back down to the resolution you actually care about, you'll end up with tremendously more SNR that way than if you follow the conventional routine. And if we go back and sort of look at those classical MR papers that argued, hey, don't go for a higher resolution, they're not quantifying resolution in the correct way. They're not looking at point spread functions. They're quantifying resolution by voxels, number of voxels, which is not the right way to measure resolution. So we have sort of all of these aspects rolled into how this works. So maybe that's too much math. Let's look at some applications of how this works. This framework can be used in a lot of different applications. In these images on the left here, I'm showing data that was acquired at 9.4 Tesla. So this is really high field MR data. This is from the University of Illinois at Chicago, Keith Felborn's group. They were doing 17.0 data acquisition. So this is naturally abundant oxygen-17. Oxygen-17 is not very naturally abundant. And so even at this high field strength of 9.4 Tesla, you still get very, very noisy looking images. And of course, you want images that look better than that. One of the things you can do at this field strength is you can get really clean looking sodium images. And within our framework, if we use these sodium images to constrain the reconstruction of our oxygen-17 images, we can get images that look much, much cleaner. And if you go and do some of the quantitative bioscales associated with this, we're getting much more accurate numbers from this set of reconstructed images than we would have from the noisier data set. We can do this in other applications as well. So this is an example of spectroscopic imaging of the mouse brain. This mouse has been given a stroke, and you can see the stroke lesion there in the image. We're trying to do spectroscopic imaging here so we can really see the metabolite distribution in the brain. This is 14 Tesla data that was acquired at Washington University in St. Louis. You can see here that our reconstruction of N-acetyl L-aspartate, so this is sort of a marker of the viability of neuronal mitochondria. This looks fairly noisy, but if we have sort of separate proton anatomical reference images, we can get really clean maps of NAA here and lactate with super high resolution for this type of application. And we can see very cleanly we have an increase of lactate in that spatial position where the blood supply has been cut off, which makes a huge amount of sense when you think about the pathology. So we can also use this in diffusion. When we apply it to diffusion, we get major improvements in tractography quality. We can apply this in animal imaging experiments where a typical acquisition might have lasted 2.5 hours. We can now get exactly the same quality quantitative metrics in about 40 minutes of data acquisition. That makes a huge difference when you're trying to keep these animals happy and alive in the scanner. And when you look at human data, you also are able to get very strong improvements in image quality just by doing some extra signal processing at the end. We've been recently trying to push this idea to the extreme, and this is joint work we're doing with Colin Setzenpop's group at MGH, where we're trying to do in vivo brain imaging at really high resolution. So this is 660 micron isotropic resolution data I'm going to be showing you here. Our mathematical formulation is quite complicated. We're doing some interesting things with the physics as well that I'm not going to go into. But let me just show you the type of result that we're able to get. So this, again, is 660 micron isotropic resolution diffusion weighted imaging, relatively high B-value, not super high, but higher than most acquisitions. And if you spend 25 minutes to get 64 directions plus 7 B-values, this is what one of your diffusion weighted images looks like. You can see a lot of structure present there, but you can also see a lot of noise that's present there. But if we use the fact that these images are not independent from each other, we can substantially clean up this noise. And when we go and compute quantitative parameters from that, we get these really, really nice high resolution maps of all the structures that are present in the body, or in the brain here. This is 3 Tesla data, 660 micron in 25 minutes. This is a result we presented in 2016. We've been able to show that we've been able to go from 25 minutes to 15 minutes. That's not a problem. One thing I didn't mention, but I should mention now, that this data was acquired on the Connectome scanner at MGH. This is a special scanner that has really nice gradients that most people don't have. But very recently, we've moved this to a standard commercial 3T scanner. And we've been able to show that in 10 minutes, we can get 860 micron resolution, really high quality images using this type of technique. And one thing I forgot to mention, this difference here, this is about the same as if I had averaged this data 3 times. So you can think of, we're getting the quality of a 75-minute scan from a 25-minute scan by doing this. Okay, and I mentioned to you before that not only are we able to get nice results like this, but we actually have a strong theory that tells us exactly how we want to be making these trade-offs. It influences the way we do data acquisition, et cetera. Here's an example where we've done standard reconstruction of this mouse brain image. We know the resolution that's associated with that standard reconstruction. If we use our constrained reconstruction technique, we can control it so that we have exactly 4 times noise variance improvement. That's an improvement in imaging time of a factor of 4, since you no longer need to do that same amount of averaging. We've lost a little bit of resolution, but not as much as you probably would have thought. And we can go to 8 times noise variance reduction, and again, the loss in spatial resolution is there. It's present. We're not pretending that this is a perfect representation of the original high resolution image, but the compromise we've made here is not as big as what you would have predicted initially. And this really tells you, well, I should go for higher resolution. And then use this type of technique to get rid of my noise, rather than just spending all my time averaging low resolution data. Okay, so that was the first example I was going to talk about. I'll also talk about sparsity constraints. Sparsity constraints are probably the most widely known type of constrained imaging method. The basic idea here is that if we take an original MR image, and if we do a wavelet transform or a discrete cosine transform, these are sort of the transforms that get used in things like JPEG compression. We know that MR images are not that different from other types of images that you might take with your digital camera. You'll end up with a lot of sparsity. That is, in this transform domain, a lot of the coefficients have small values. And because they have small values, you can throw them away, and you don't make big compromises in image quality by throwing those away. So this leads to compression. MR images are also smooth, so if you take local differences between neighboring pixels, you'll also see sparsity characteristics there. So the fact that MR images are powerful, that they're sort of compressible in these transform domains, gives us the hope that we don't have to acquire all of that data to begin with if we know that we can just throw a lot of it away in the end. So the basic compressed sensing idea is don't sample based on the number of voxels, which is what the Nyquist rate would tell you to do. Instead, sample in proportion to the sparsity. This is a really old idea, and if we look at some of the earliest work, which is from the 80s, the idea there was, hey, MR images are sparse, so let me acquire low resolution data in the Fourier domain and use the fact that these images are sparse to achieve super resolution. Let me use that information to sort of extrapolate the high frequency information I didn't acquire and get nice looking images as a result. So this was a very early idea, but it didn't catch on that well. Why didn't it catch on? It turns out you can do this, but it's very sensitive to noise. And we have noise in MR, and so extrapolating over long distances gives you very, very noisy results. And so this didn't really catch on in a big way. Later, we have a lot of buzz associated with compressed sensing theory and the math and engineering worlds arising. And this led to a different way of acquiring data, what we usually call compressed sensing these days, where instead of just acquiring low resolution data and trying to extrapolate, instead we're sort of randomly sampling across, whoops, hope I didn't break anything. So we're just sort of randomly sampling the Fourier space here, and then using sparsity to sort of interpolate some of that missing information. And interpolation, it turns out, is a lot easier to do than extrapolation. The distances involved in interpolation are much smaller, and so you don't have the same robustness issues that you do when you're trying to do extrapolation. But I was looking at this and felt a little bit unsatisfied with this random sampling paradigm. And I was unsatisfied because when you look at some of this math literature, it makes the argument that certain types of randomness in acquisition are good. But when you start really looking at the details, the type of random Fourier sampling that we commonly use in MR, this is not the type of randomness that they were prescribing in the math literature. And so there are a number of criticisms I can make about random Fourier sampling here. I'm not going to read through all of these for you, but I was interested in exploring other ways of relying on sparsity. So we have this model, sparsity is our model. How can we do data acquisition differently in a way that best exploits the fact that these images are sparse? And so the first thing I thought about in this regime was, well, this idea of Fourier encoding when I have a sparse image is not really the right way to do things mathematically. If you look at the mathematical literature, they like a different type of randomness. And I knew enough MR physics to know that we didn't actually have to do Fourier MR. So Fourier MR is the most common approach, is the prevailing approach. So if you're using a commercial scanner, you're definitely using Fourier MR, but it's not what you have to do. And in particular, if you replace your normal slice selective excitation pulse with something that has some random characteristics to it, you can acquire an image that has random magnitude modulations and random phase modulations associated with it. And if I do a different RF pulse at every TR, I can get much, much closer to this sort of random scheme that is more theoretically optimal when I look at the mathematical literature. And so we went ahead and implemented this. This is something we implemented at 14 Tesla at the University of Illinois. We can see that if we compare to random Fourier sampling, we're able to get much higher quality images with much less data using this random type of RF encoding. And so this was nice, this was good progress, but there's a reason we sort of stopped at 14 Tesla as well. Translating this to a human application faces a number of different obstacles and is not the easiest thing to do. So, but this already sort of reinforced this idea for us that random Fourier sampling paradigm that most people had been using in the field was not necessarily the right way to proceed with this kind of data. The next question we asked, is, well, okay, we know that random Fourier sampling is not necessarily the best. Can we do this in a deterministic way? Do we have to sort of treat this problem as having the good luck to get a good solution? Luck here, remember, randomness is not as random as you might initially expect. So you can have a good amount of luck, but we wanted to do things deterministically. And we wanted to do things that were application specific and hardware specific. And it turns out that it is possible to do this. Again, I'm not going to go through the details, but you're free to ask me about it later if you want to. We have a TMI paper on this that is out right now. We call our framework Oedipus just because we like having nice acronyms in MR. But the basic idea is that if I give you a specific image and I give you a specific type of hardware configuration, maybe I have a single channel coil, maybe I have a 12 channel coil, maybe I have a 32 channel coil, etc. We have a deterministic algorithm which will tell us how we want to sample that data for that specific image. And you can see here that between single channel and 12 channel, we're getting adaptation. Right? It's not the same thing. Between single channel and 12 channel, we're getting differences. And people in the community already knew that you should sample K-space in different ways. You should sample the Fourier space in different ways in these different contexts. But it was largely heuristic, the methods that were used to design exactly how you tune all these different parameters. Now we have a deterministic way of doing that. So let me very quickly go through my third point here on low rank constraints. This is sort of historically interesting, and it precedes my final one in a nice way. So I'll go through this quickly. The basic idea here is that if you look at a dynamic image, an image that changes over time, it turns out that if you take this data and rearrange it into a matrix where one dimension is spatial, the other dimension is temporal, and you do a principal component analysis of that image, it turns out that in almost every case you don't get a huge number of important principal components. There's a lot of redundancy in that data, a lot of components you can just throw away without really losing any of the key features of that image. I'm showing an example down here. This is not a dynamic scan, this is a scan where we're changing acquisition parameters to encode different relaxation parameters here. This image is well approximated as having 4 different components, 4 different principal components. This representation is nice because if we only think about 4 components, that's 29% of the degrees of freedom. That means we can undersample the acquisition of this data by a tremendous amount if we want to rely on this low rank model. Now, the fact that it's low rank is also nice because there's a lot of different theory you can develop and a lot of different algorithms you can develop to recover low rank matrices from limited samples. And I'm going to skip all of that and just sort of show you this one example here of a beating rat heart. This is real-time, ungated, free-breathing rat cardiac imaging. It's been slowed down so you can see what's going on, but the TR here is 7.5 milliseconds. If you just do standard acquisition, you can't really get to high spatiotemporal resolution because things are changing so quickly in time. And so here we're violating the temporal Nyquist rate, and you see smearing of the temporal dynamics. But by imposing this low-rank matrix structure, we're able to get really nice depictions of what's going on at high speed in that rat. Okay, so that leads us into the next topic, which I've been doing a lot of work on in recent years, which is based on low-rank linear predictability. So it turns out that if you look at MR data, the Fourier data in MR is autoregressive. And there are a number of different ways you can show that the data is autoregressive. Colin Sessenpop and I have a review article on this that you can find on the archive right now. But the basic idea of autoregression is that if you are missing a data sample, you can usually predict that missing data sample from a linear combination of its neighbors. The same idea gets used in your cell phones all the time for speech coding. That rather than sending your entire speech audio signal across the network, your cell phone is going to say, hey, your audio signal is linear predictable. Let me find out the linear predictive coefficients that I need to send across the network for people to reliably reconstruct the sound at the other end without sending quite as much data. An important aspect of this, though, is that you need to figure out what that prediction relationship actually is in order to exploit this. So, as with a lot of things in MR, linear predictability is an old idea. It was shown in the 90s that if your image is support limited, that means you have zeros in your field of view. You can show that you have linear predictability in the Fourier domain. It turns out that if you have an image with smooth phase, and a lot of MR images do have smooth phase, you can linearly predict one sample from a set of samples on the opposite side of the Fourier domain. Recently, we've shown that you can also do this linear prediction using data on both sides. If you have multi-channel images, and this is very, very common in modern MR, that you're acquiring data with an array of receiver coils that each see a slightly different view of the same image, you have multi-channel linear predictability. This is also true, by the way, if you have different contrasts of the same image. You can also get linear predictability across those. And then that early sparsity example I showed you before, that's also based on linear predictability. If your image is sparse in a transform domain, then you can do autoregression in that transform domain. So all of that is nice. It shows us we have the potential to use linear predictability, but how do we actually do it? So for the past several years, we've been developing this Lorax framework that sort of takes all of these constraints into account, combines them into one. It automatically identifies whenever autoregression is present in your data and exploits that as much as it can. And it does this by using a structured low-rank matrix model. So the idea here is that you have your original Fourier data, and you're taking sort of local neighborhoods of that data and forming that into this really, really redundant matrix. What we can show is that in the presence of this autoregressivity, these low-rank matrices we're constructing have to have low rank. So there's going to be null space vectors associated with our linear prediction relationships. And just like before, we can reconstruct low-rank matrices from undersampled data. So we're exploiting that here. Compared to the previous low-rank example I showed you, here we also have an additional blessing of dimensionality, that we're starting with something relatively low dimensional and mapping it into higher dimensions where it has even simpler structure than it previously would have, and we can definitely exploit that. So let me just give you some quick examples of how this works. Here is an MP RAGE image, a high-resolution T1 weighted image. If you fully sample the data, maybe it takes 11 minutes. Given this new framework we can do sampling in ways that no one ever dreamed was possible before. So here we've sampled according to the USC logo. I don't recommend this, by the way, but we're just doing it to show that it's possible. If you do traditional sparsity-based reconstruction, you get garbage, and you'd expect to get garbage because this is not a good way to sample data. But if you use our Lorax framework, this is not a perfect image, but it's much better than what you would have expected. Now, if you actually use this in a good way, so in this case we're doing wave Kuiper acquisition, which is already, by the way, a state-of-the-art type of approach. We're able to get to 16 times acceleration. So this is about a 40-second MP RAGE acquisition. And we have higher quality than the previous state-of-the-art, which was only 9 times accelerated. So there's a lot of open questions here. There's a lot left to be explored, but we're getting some pretty neat results with this so far. So before wrapping up, I want to say something about high-dimensional MR again. So if you remember my earlier slide, we talked about sort of this dream of getting a 12-dimensional experiment that tells us everything we want at once. We can separate everything in the ways that we want. We haven't quite gotten to 12 dimensions yet, but I'll very quickly show you some of the lower dimensional things that we've done. So my students have been working on 2 techniques so far. One is called diffusion-relaxation correlation spectroscopic imaging. Another is called relaxation correlation spectroscopic imaging, DRCSI and RRCSI. In these cases, we're trying to do high-dimensional diffusion and relaxation encoding or high-dimensional relaxation encoding. And here is just a quick example of a 4-dimensional DRCSI experiment where we have diffusion encoding along this dimension, relaxation, T2 relaxation encoding along this other dimension. Now from that data, we're able to reconstruct a spectroscopic image. And I know this is probably going to be hard to see here, but at every voxel, we have a spectrum. The spectrum tells me what the diffusion and relaxation characteristics are like locally within that voxel. And let me zoom in a little bit here. So this is the boundary between white matter and gray matter in this mouse spinal cord here. Might be rat, actually. But what you can see, we have peaks for the different microstructural features of the tissue that are present. And at the boundary, we very, very clearly see the partial voluming occurring. So we're able to reliably separate out that partial voluming using this type of technique. Now, this is a healthy cord. Well, it's outside the animal, but nothing what was done to the cord while the animal was alive. But it gets more interesting when you start looking at pathological cases. So in this case, we have an injured cord, traumatic injury, that there was a laminectomy performed, and basically the cord was hit with a hammer in a controlled type of way. We actually see very different spectra between the control cord versus the injured cord. And if we separate out the different spectral peaks we see, we see nice white matter characteristics, gray matter characteristics, additional gray-white types of characteristics. But then there's this component 4 here, which only shows up in the injured cord. And so we're seeing some kind of pathology manifesting here. We haven't had the chance to do histology of this to really pinpoint what it is we're seeing here, but we appear to be seeing features of this tissue that we haven't seen with other types of techniques. We can use the same type of idea with T1 relaxation and T2 relaxation. Here's some human brain examples, and really sort of the first human in vivo examples of this type of high-dimensional imaging technique, where we're able to see lots and lots of different components. The components make sense anatomically, and these are all healthy subjects. It'll be really interesting once we start playing around with this in subjects with pathology. Okay, so to sort of summarize what we've been through in this talk, MRI, as we all know, has had a tremendous impact so far. It's transformative across a wide variety of fields, but even though it's been developing for so many decades, it's still not mature. There are things that Paul Lauterbur imagined back in the 70s that we still haven't been able to do efficiently, and there's a potential to do much more. One of the things I hope I've convinced you of is that if you combine signal processing ideas with the blessings of dimensionality, you can achieve faster and better MR, and there's a large number of interesting theoretical results and methods and applications that go along with that. Just to wrap up, I like this quote by Erwin Hahn, that there's nothing that nuclear spins will not do for you as long as you treat them as human beings. I think if you look at the news right now, you'll see that we still don't quite know how to treat human beings, and so that there's a tremendous amount left to do here as well, I think. So with that, I'd like to thank you for your attention, and I'm happy to take any questions.
Questions?
So a number of these techniques obviously are enormously beneficial in terms of accelerating MRI acquisition and secondarily also increasing signal-to-noise resolution. How close are they to, say, practical / commercial application?
So it's a good question. Some of these things are deployable, I would say. So some of these things we're packaging and people are routinely using at our Neuroscience Imaging Center, for example. Some of these things our collaborators are routinely using. And so it's available if you're willing to go through some of the hurdles to make everything smooth. In terms of getting it onto a commercial scanner, a lot of that is about workflow and user interface. And those are aspects that are important, but they're not as much aspects that I'm interested in spending a lot of my time on. I sort of like working on the math and showing things are possible. And then if ideas gain enough traction, then someone in the community will make that widely available. So for a lot of the techniques we're showing, one of the bottlenecks right now is how much time it takes to do the reconstruction. And depending on what your environment is, that can be either a bottleneck or it's fine. And beyond that, it's really a lot of workflow things. What is the data format? How do you get the data you need off the scanner? How do you send that to the right pipeline so that it gets reconstructed in the right way to get fit back as needed by your specific application? And so that can be very application dependent. But I would view those as sort of the large bottlenecks for practical translation. That there's another level of it in terms of FDA approval and things like that, that is also an obstacle.
So in the shared edge constraint model, can you quantify the leverage that each measurement is bringing to the final solution? So in other words, how is your solution being affected by?
Well, so what we have in that case is for every spatial position in my original image. And remember, the original image here is not actually discrete in terms of a finite number of voxels. The original image is continuous. So for every point in that original image, we have a mapping from that point to the reconstructed image. And so we basically have a point spread function. This is not a shift invariant system, so we'll call it a spatial response function rather than a point spread function. But we have this direct mapping. So we understand that very well.
To what extent can you deal with like the cross-session in exploiting this? Say you're interested in changes in anatomy. Could you have one image that you started with and then have follow-up images be much sparser and yet capture the changes in the presence of head movement in different positions?
Yeah, so that's a very good question. How much can you exploit redundancy across sessions? There are ways of doing that, but it tends to be a little bit more challenging than when you're doing it within the same session, because you don't exactly know what has changed. You know that some things probably have changed, but you usually need to do some kind of registration procedure. A lot of the sort of underlying physical characteristics of the image, the image phase, the sensitivity profiles and everything, those will all have changed between sessions. And so you need to come up with a formulation that ignores some of those changes. People have been fairly successful doing this type of thing with interventional procedures, where they're doing surgery inside the scanner, maybe moving a catheter around inside, and all kinds of things are moving around. But it takes some finesse to get that to work properly. And of course, in medicine, you generally want to be conservative, right? And if you rely too much on prior information, you might end up with the wrong image as the result. And so right now, sort of longitudinally using this is maybe a riskier proposition in terms of how much confidence you can have in your final result compared to doing it within the same session. Yeah?
So when you, sometimes you see papers and they show, here's my gold standard and then here's sped up, and they look identical.
Mm-hmm.
And we're kind of stuck there a little bit, I think. Do you have any kind of more quantitative or suggestions how you can show the trade-off that you're making?
So it depends a lot on the way you formulated your problem. So in the shared edge structure example I showed, in the first example I showed, we there had a very, very clear trade-off because we had access to these spatial response functions. We know what our resolution trade-off is. We know what our signal-to-noise trade-off is there. Because everything was linear enough. It wasn't linear, but it's linear enough that we can make accurate predictions with respect to those things. And so that gives you a very high standard to compare things against. For some of these other methods using sparsity or low rank structure, these are much more highly nonlinear. And so that level of characterization is still something we don't have a good grasp of right now. The field in general doesn't have a good grasp of how to do that. There are things you can do in simulation that go beyond just, hey, look at my two images, they look the same. So if you have a gold standard, you can compute an error image with your reconstruction, and there are a variety of quantitative measures you can look at. A mean squared error is fairly common. There are measures of visual, perceptual similarity. And one of my students recently came up with this error spectrum plot where rather than reducing this to a single number, you can actually say, look at the errors as a spectrum and say, at low resolution we're doing pretty good, but at high resolution frequencies we're starting to get a lot of errors. Those types of techniques also have their own limitations, though, because it requires you to have a fully sampled gold standard to compare against. And when you're actually implementing some of these sequences, a variety of practical things start to come into play that wouldn't be captured by a simulation from retrospectively undersampled data. So if I'm doing an experiment that has an echo train, I'm going to get relaxation during that echo train, and that's not going to be captured by my simulation in an accurate way. So there's still a lot of room for improvement, but we can definitely go beyond just visual assessment.
Any other questions? So I had one last question. So there was a lot of talk about non-local properties, but that was, so most of that work was being applied to the image domain, not in the case spaces, or I know. Is there something going on in that direction these days, or that's?
So non-local in K-space?
Yeah. Properties, statistics, and.
Well, it depends on how you want to define non-local.
Yes. So we can do it in the image space well.
Yeah. So people aren't doing things like non-local means, for example, in the Fourier domain. And it would be hard to apply something like non-local means in the Fourier domain because the Fourier domain has different characteristics than the image domain does. The data decays in a specific way, which makes it harder to find similar K-space patches. The complex valued nature also makes it a little bit more challenging. But in some sense, the Lorax approach I've described here, because it's shift invariant,
so
it's both local and sort of non-local, because this is a consistent characteristic that all of these patches in K-space will share. So you could view it that way.
Cool. So let's thank Justin again for that. and see you next time. Very nice. Very nice
Thank you.
talk.